deoxyribose molecule. That is a deoxyribose molecule, as well as it is made up of the deoxyribose molecule, as well as the phosphate molecule, whereas the steps of the ladder are made up of a pair of nitrogenous bases. So there are four different types of nitrogenous bases, that is adenine, thymine, guanine, and cytosine. And the nitrogenous bases have got a specific pairing, or the pairing between these nitrogenous bases is always specific, because here the amount of adenine equals to that of the thymine, and the amount of guanine equals to that of the cytosine. So always adenine pairs with thymine by 2 hydrogen bonds, whereas guanine pairs with cytosine by 3 hydrogen bonds. See here, if you see the structure of the DNA molecule, see this is a double helical structure. The nitrogenous bases are adenine, thymine, guanine, and cytosine. So this is actually, these are the sides and these are the steps of the ladder. So you have a major groove as well as a minor groove is there. So on the sides, you have got a sugar phosphate backbone. That means it is made up of sugar plus the phosphate, whereas the steps actually are made up of the nitrogenous bases. So this is how it looks like. So like DNA, RNA, it consists of 3' end as well as the 5' end. So this is the structure of the DNA. Now the chemical structure of the DNA, same way here also they have same given. See here, if you see the sugar phosphate backbone, this is so adenine, thymine, guanine, and as well as a cytosine. So double helical structure, it just looks like a twisted ladder, and what holds the DNA stands together. Okay, so from DNA to the proteins. So DNA then it will form the RNA, and the RNA will form the protein. So that is how this will, I'll tell you when we go to the protein synthesis. Okay. Now coming to the chemical structure of the DNA. So it is a double stranded helix or a double helical structure. That is, each DNA molecule actually is comprised of what? 2 bipolymer strands coiling around each other to form a double helical structure. So that means each DNA molecule consists of 2 polymer strands which coil around each other, forming a double helix. And these 2 strands actually are called as the polynucleotides, as they are made up of the simple monomeric units, which are called as nothing but the nucleotides. So that means each DNA molecule actually consists of a number of polynucleotides. And each strand, if you look at it, it has got a 5' end with a phosphate group and a 3' end with a hydroxyl group. Then these strands actually are antiparallel to each other. That means one strand will run in a 5' direction, a 5' to 3' direction, while the other strand will run in opposite way. That is 3' to 5', 3' to 5' direction. 3' to 5' direction. And the 2 strands are actually held together by the hydrogen bond, and they're complementary to each other. And the DNA, you all know, actually basically it is composed of the deoxyribonucleotides, and these deoxyribonucleotides again are held together by means of 3' to 5' phosphodiester bonds. And the nitrogenous bases that compose these deoxyribonucleotides, you all know, are adenine, cytosine, thymine, as well as the guanine. And the complementarity of these strands, it is nothing but due to the nature of the nitrogenous bases. That means the base adenine always interacts with the thymine by 2 hydrogen bonds, whereas cytosine always interacts with guanine by 3 hydrogen bonds. And the shape of the helix actually is being stabilized by the hydrogen bonding as well as the hydrophobic interactions between these nitrogenous bases. And the diameter of the double helix is 2 nanometer, and the double helical structure repeats at an interval of each interval of about 3.4 nanometers, which corresponds to 10 base pairs. So that is the double helical structure of the DNA, which was given by the Watson and Crick. So as given by Watson and Crick, DNA actually is a double stranded helix, or you can say it has got a double helical structure. That means each DNA molecule actually has got or consists of 2 biopolymer strands which coil around each other to form the double helical structure. And these 2 strands actually are called as the polynucleotides, as they are made up of a number of monomer units which are called as the nucleotide. And each strand actually has got a 5' end with a phosphate group and a 3' end it has got a 5' end with a phosphate group and a 3' end with a hydroxyl group. And these 2 strands are actually anti-parallel with each other. That means one strand always runs in 5' to 3' direction, and the other one will run in a 3' to 5' direction. And these 2 strands are held together by the hydrogen bonds, as I already told you. And the DNA is made up of composed of deoxyribonucleotides, nucleotides which are held together by 3' and 5' phosphodiester bonds. And the nitrogenous bases that compose these deoxyribonucleotides are nothing but they are adenine, thymine, cytosine, and guanine. And these strands are complementary to each other. That means always adenine pairs with thymine by 2 hydrogen bonds and cytosine pairs with guanine by 3 hydrogen bonds. Shape of the helix is being stabilized by the hydrogen bonds as well as the hydrophobic bonding. And the diameter of the double helix actually is 2 nanometer, and the double helical structure repeats at an interval of 3.4 nanometer, which corresponds to 10 base pairs. So this is the double helical structure which was given by the Watson and Crick. Now, coming to now we will see what are the major and the minor grooves present in the DNA. So now we have understood the actually what is the basic structure of a DNA molecule, which is nothing but a double helical structure. So we say it has got two polymer strands and all those things. Now we'll see what do we mean by major group and the minor group that is actually observed in the double helical structure of DNA. So what happens is, as a result of the double helical nature of the DNA, the molecule has got two asymmetric groups. They have got two asymmetric groups, means they are not equal together. One group is smaller than the other. One group is smaller than the other. And this asymmetry actually is nothing but it is the result of the geometrical configuration of the bonds between the phosphate sugar and the base groups that forces the base groups to attach at 120-degree angles instead of 180-degree. Okay, so as a result of the double helical nature of the DNA, this molecule actually has got two asymmetric groups. One group is smaller than the other, or you can say one group is bigger and the other one is a smaller. And this asymmetry occurs due to the result of the geometrical configuration of the bonds which is present between the phosphate sugar as well as the base pairs. And the larger group actually you call it as a major group, which occurs when the backbones are far apart, and the smaller one actually is called the minor group, which occurs when they are very close to each other. So these groups, one we call it as a major group and the other one we are calling it as a minor group. The larger one we are calling it as a major group because here and this occurs when the backbones are very far apart, whereas when the backbone are very close to each other, then the smaller group, that is a minor group, will appear. Since the major and the minor groups expose the edges of the bases, then the groups can be used to tell the base sequence of a specific DNA molecule. And the possibility for such recognition is critical since the proteins must be able to recognize the specific DNA sequences on which to bind in order for the proper functioning of the body as well as the cell to be carried out. So this is about the major as well as the minor groups of the DNA molecule. Now coming to the properties of the DNA. So what are the basic properties of the DNA? Now you all know DNA helices can be right-handed or left-handed. So that means you have got a right-handed DNA as well as a left-handed DNA. So it can be either right-handed or it can be either left-handed. So DNA having the right-handed helices is the most stable one. So the DNA molecule which holds or which has got the right-handed helix actually is preferred to be the most stable one. So on heating, what happens is the two strands of the DNA, they separate from each other, and on cooling, they again will come close together and hybridize. Okay, so the two DNA helices may be right-handed or left-handed, and the DNA which is having the right-handed actually is the most stable one. So on heating, what happens? The two strands of the DNA can be separated from each other, and on cooling, again the two strands will come close together and try to hybridize. And the temperature at which the two strands separate completely is known as the melting temperature. So at the melting temperature, actually what happens is the two DNA strands will separate, and the melting temperature is specific for each specific sequence. It is specific for each specific sequence. And the B sample of DNA having higher melting point must have more CG content because CG bonds. The sequence of bases along the DNA molecule encodes for the sequence of amino acid in every position. So okay, so we can say that the major properties of the DNA is nothing but it can be either right-handed or it can be left-handed, but the B configuration of the DNA having the right-handed is the most stable. Why? Because here in this case, why we are saying is cytosine will pair with guanine and it has got 3 hydrogen bonds. Whereas the A pair actually, their adenine will pair with the thymine only by 2 hydrogen bonds. So that is why actually they are saying that the B configuration DNA having the right-handed helix is the most stable. Okay. Now when compared to the A, we can say. So on heating, what happens is the two strands of DNA will separate from each other, and on cooling again they will come close together and try to hybridize. And the temperature at which these two stand separate completely is called as a melting temperature. And this melting temperature is specific for each specific, the sequence of the DNA. And the B sample of DNA having higher melting point must have higher, more CG content. So this is about the properties of DNA. Now coming to types of DNA. Now, as we have already, I have discussed yesterday regarding the types of RNA. So here also we find different types of DNA are also being present. Now we'll see now what are the different types of DNA. So you all know actually eukaryotic organisms like plants, animals, fungi, they store the majority of their DNA inside the cell nucleus and some of their DNA in the organs such as mitochondria, So based on their location, they have been classified as nuclear DNA as well as a mitochondrial DNA. Okay, so majority of the eukaryotic organisms, basically actually DNA is present in the nucleus itself, but some amount of DNA you also find it in the mitochondria, very little amount. So that is why it is called as a mitochondrial DNA. So based on the location of this DNA, the DNA have been classified into two different types. What are those? One is called nuclear DNA and the other one we call it as a mitochondrial DNA. Now we'll see what are the differences between these two. Now coming to the nuclear DNA actually is located within the nucleus of the eukaryotic cells. It is located within the nucleus of the eukaryotic cells. Now usually it has 2 copies per cell. The nuclear DNA located within the nucleus of the eukaryotic cell, it usually has 2 copies per cell, and the structure of the nuclear DNA chromosome is linear with open ends, and it includes 46 chromosomes containing 3 billion nucleotides. And the nuclear DNA is always diploid, inheriting the DNA from the 2 parents. And the mutation rate for nuclear DNA is less than 0.3%. Okay, so this is about the nuclear DNA. Where is nuclear DNA located? It is located within the nucleus. It has 2 copies per cell, and the structure of this nuclear DNA is linear, containing how much? 46 chromosomes with 3 billion nucleotides, and it is always diploid, inheriting the DNA from the 2 parent cells. And the mutation rate for this is less than 0.3%. Now coming to the mitochondrial DNA, it is located in the mitochondria. It contains 100 to 1000 copies per cell. So it has more, so 100 to 1000 copies per cell. And mitochondrial DNA chromosomes usually have closed circular structures. They have closed circular structures and they contain about 16,569 nucleotides. How much? 16,500. So less number of nucleotides it contains. And the mitochondrial DNA, basically it is haploid, coming only from the mother. It comes only from the mother and it is haploid. And the mutation rate is very high when compared to that of the nuclear DNA. So this is the difference between the nuclear DNA as well as the mitochondrial DNA. Okay. Now, next is most of the DNA actually is in the classic Watson-Crick model, simply called as the B-DNA or the B-form DNA. Okay, so most of the DNA actually is present in the it holds, or you can say it adjusts to the Watson-Crick model. So that is why it can be called as a B-DNA or the B form of DNA. In certain conditions, different forms of DNAs are also found, and they are found to be appeared like A-DNA, Z-DNA, as well as B-DNA. So this deviation actually informs is based on their structural diversity. It is based on their structural diversity. So basically, majority of the DNA, if you observe, they all actually are the B form of DNA, which holds or which actually tries to, agree with the Watson-Crick model. But in majority of the conditions, different forms of DNA are being observed, like you have A-DNA, Z-DNA, as well as the B-DNA, and these DNA actually occur due to their deviation in the structural diversity. Now we'll see what do you mean by B-DNA, A-DNA, as well as Z-DNA. B-DNA actually is the most common. It is originally deduced from X-ray diffraction of sodium salt of DNA fibers at 72% relative humidity. And A-DNA actually was identified by X-ray diffraction of analysis of DNA fibers at 75%. Whereas Z-DNA actually is called as a left-handed double helical structure, which binds to the left in a zigzag pattern. So that is how they have identified the other forms of DNA. So basically, we have how many? 4 types of DNA we can say. But if you see in general, you have only 2 types. That is called as a right-handed as well as a left-handed. Now, right-handed is the B-DNA, whereas left-handed actually is what we call it as the Z-DNA. We call it as a Z-DNA. Okay, so Watson and Crick model, it's called as the BDNA. So here, apart from these two, now we'll see what is the differences between these. So these DNA. So you have ADNA, you have, then you have BDNA, cDNA, as well as the ZDNA. Okay, so ADNA, then you have got the BDNA, then you have got the cDNA, and then you have got the ZDNA. So these are the different types of DNA actually that you find. Okay. Now, coming to the functions of the DNA. Now, what are the different functions of the DNA? For what purpose actually DNA is being present? So you all know actually DNA has a very crucial role to play because it is a genetic material in most of the living organisms. So it has to carry the information or the message from cell to cell and from generation to generation. So the major functions includes what? Storing the genetic information, then directing the protein synthesis, then it also has to determine the genetic coding. It is also responsible for various metabolic activities, evolution, heredity, as well as differentiation. And it is a stable molecule and it holds more complex information for longer periods of the time. Okay, so this is all about the functions of the DNA. Okay, now, so with this, now we have come, to the conclusion of the DNA structure. Now, coming to the DNA structure, again, we will brush up. So, you all know DNA, like RNA, it is also one of the nucleic acid. It is very, very important, and it is one of the major molecules that is essential for a cell, for all the forms of life. Actually, it is found in the nucleus with a small amount of DNA also being present in the mitochondria, which is called as the mitochondrial nucleus. And actually, in the year 1953, Sir James Watson and Francis Crick discovered the structure of the DNA, and they called it as a double helical structure. Later on, the scientist by name Rosalind Franklin also carried out some work, and according to his view, he pointed out that the DNA is made up of two spirals, or it is made up of a double helical structure. It is made up of a two spirals. And the structure of the DNA actually is a double helical structure. Because why we are calling it as a double helical structure? Because it just looks like a twisted ladder, you can say, and where the sides of the ladder you have, and you have got the steps of the ladder. The sides of the ladder are made up of alternating sugar as well as the phosphate molecule, while the steps are made up of a pair of nitrogenous bases. And like RNA nitrogenous bases, like adenine, thymine, guanine, and cytosine. Now, only difference between these two is in case of DNA, you have thymine, but in case of this thymine RNA, thymine is being replaced by uracil. And the nitrogen bases have a specific pairing pattern over here, because the pairing pattern always occurs between the adenine as well as the thymine by two hydrogen bonds, and guanine with cytosine by three hydrogen bonds. So this is the structure we have already seen. So it is a double helical structure. You all know, and each DNA molecule actually is made up of two biopolymer strands which coil around each other to form a double helical structure. And these two strands required as a polynucleotide, because they are made up of a number of simple mononuclear units called as a nucleotide. And each strand, if you observe, it has got a 5' end and a 3' end. And these strands, the two strands are anti-parallel, because one strand runs in a 5' to 3' direction, and the other strand runs in a 3' to 5' direction. And these two strands are held together by the hydrogen bonds, and also they are complementary to each other. They are complementary to each other. And this is all about. So the shape of the helix actually is nothing but because of the stabilization of the hydrogen bonding as well as the hydrophobic interactions. And the diameter of the double helix is 2 nm, and the double helical structure actually repeats at an angle or at an interval of 3.4 nanometer, which corresponds to 10 base pairs. Or you can say at an interval of each 3.4 nanometer, you find how many base pairs? 10 base pairs are being present. So this is how the double helical structure of the DNA molecule. Now, when you observe the structure of a DNA keenly, you find two types of grooves. One is called as a major groove, and the other one you call it as a minor groove. The one groove, so as a result of the double helical nature, the new molecule has two asymmetries. One is called as a major groove, and the other one is called minor groove. And this is nothing but this result due to the geometrical configuration of the phosphate sugar as well as the base groups. So the larger group, you call it as a major group, it occurs when actually the backbones are very far apart, while the smaller ones is called as a minor group, and it occurs when they are close apart. And since the major and the minor groups expose the edges of the base, the groups can be used to tell the base sequence of the specific DNA model. Now, coming to properties, the DNA helices can be either right-handed or left-handed, and the DNA, you can say B conformation DNA are always right-handed, and they are more stable when compared to the Z1, that is left-handed. On heating, the two strands of DNA will separate, and on cooling again, they will come close together and try to hybridize. So this is one. Then you have types of DNA. So we have got two types of DNA, that is nuclear DNA and mitochondrial DNA. DNA found in the nucleus, we call it as a nuclear DNA, and the DNA which is found in the mitochondria, you call it as a mitochondrial DNA. Mitochondrial DNA. In the nuclear DNA, you have two copies per cell, and the structure of the DNA is always linear with open ends and consisting of about 46 chromosomes with 3 billion nucleotides. Whereas the nuclear DNA is haploid, you can say, and mitochondrial DNA, sorry, nuclear DNA is diploid, whereas mitochondrial DNA is haploid. It is located in mitochondria, containing 100 to 1,000 copies per cell, and it usually have closed circular structure rather than the linear structure. So this is about the types we have seen. Now, coming to the different forms of DNA, these are aDNA, bDNA, cDNA, and dDNA. Most of the DNA in the classic Watson and Crick model, they actually have bDNA. And in certain conditions, different forms of DNA are also being present, that is aDNA, zDNA, and bDNA. Okay. So this is then coming to the functions of DNA. Now, DNA actually, since it is the genetic material, it plays a crucial role as a genetic material in most of the living organisms. So it carries the information not only from cell to cell, but also from generation to generation. So its major functions stand as it stores the genetic information, directs the protein synthesis, determines the genetic coding, and also responsible for various metabolic activities, evolution, heredity, as well as the differentiation. So it is a stable molecule and holds more complex information for longer periods of time. So this is all about the DNA molecule. So with this, we have finished with what is DNA, what are the different, what is the structure of the DNA, properties of DNA, different types of DNA, as well as the functions of the DNA. Now we will go for another chapter. Bye. Okay, now we will go to another chapter, that is protein synthesis. Now, what do you mean by protein synthesis? Now, coming to the protein synthesis, you all know actually proteins are very, very important. They are essential. They are very, very essential and structural part of every cell, and it controls every biological process in the form of the enzymes. So they need to be synthesized. So that is why protein is very, very important. Since the proteins are very important, since they play a role as an essential structural part of every cell and controls every biological process in the form of the enzymes. It need to be synthesized. So the production of a protein molecule, how actually the proteins are being synthesized. Now we'll see now. So actually it involves two phases or two processes. One is called as a transcription and the other one is called as a translation. Or we can say the protein molecule is being synthesized by with the help of two processes, or it occurs in two phases. One is called as a transcription and the other one we call it as a translation. Now, what do we mean by transcription? Now, the transfer of the genetic information from DNA to the messenger RNA is known as the transcription. That is, what is transcription? The transfer of the genetic information from the DNA to the messenger RNA is called as a transcription. So actually here in this case in transcription, what happens is whatever the genetic information, whatever the message that has to be transferred from the DNA to the messenger RNA molecule, it occurs with the process which is called as nothing but the transcription. So DNA is transcribed to the messenger RNA in the nucleus, and later this messenger RNA is translated into the protein in the cytoplasm. So this is actually the central dogma of protein synthesis. So that means just now I told the protein synthesis, actually why proteins are essential, why it has to be synthesized, because it is a essential part. It gives structure, and also it is a very, very important component of almost all the living organisms. So it has to be synthesized. And it is being synthesized by two processes. One is called as a transcription and the other one we are calling it as a translation. Now, what do we mean by transcription? First, the DNA has to be transcribed to the messenger RNA molecule in the nucleus, and later on, this messenger RNA has to be translated into the protein in the cytoplasm. So, this is what is nothing but a gist, you can say, or overall central dogma of the protein synthesis. Now, we'll see how actually the translation as well, transcription as well as the translation will take place step by step. Now see here. So DNA, this is a transcription and this is a translation. First, the DNA has to get involved. So DNA, with the help of the DNA replication, means it has to replicate. Okay, it has to be replicate, and then it has to form the messenger RNA. Then RNA has to be replicated with the help of translation, and then it has to be translated in the form of a protein. So this is actually a gist of. So first, so it involves what? It involves DNA, RNA, and protein. So the proteins are not formed directly. First, the DNA will get converted into the messenger RNA, and messenger RNA will get converted into the protein. So this is how actually the protein synthesis will take place. Now coming to the transcription. Now the first part of the process, you call it as nothing but a transcription. It occurs in the nucleus and it involves what? Messenger RNA being produced that is complementary to specific DNA sequence. So the first process, you call it as transcription. It occurs in the nucleus and it involves what? The messenger RNA molecule which is being produced and it is complementary to the specific DNA sequence. Next is translation. The second part is called as a translation. It occurs in the cytoplasm and it results in the synthesis of a specific protein molecule from the genetic information that is being carried out by the messenger RNA tool. See here, this is how transcription. First process is transcription and the second process is the translation. So that is how it involves transcription as well as a translation. Now, for transcription to take place, what are the different components that are required? One, you require a template, you require activated precursors, and you require RNA polymerase. So with this, actually, the different components, these are the different components actually that help in the process of the transcription. So what are those? One is called as a template, then you have activated precursors, then you have got the RNA polymerase. Now, what is a template? A template actually is a double-stranded DNA. Out of the two strands, only one strand is being transcribed. So template basically is a double-stranded DNA. So here, usually what happens is a double-stranded DNA out of these two strands, only one strand actually will get transcribed, not two strands, because you all know actually DNA has a two strands. So out of these two strands, only one strand actually will get transcribed. This is how. So the template DNA strand is called as a, so here the sense strand is the one that has been run. So this is how actually the template is being formed. The template strand is called as a transcribed strand with antisense sequence and the messenger RNA transcript. Then you have activated precursors. Now, what are the activated precursors? You require 4 ribonucleoside triphosphates. You require how many precursors? 4 ribonucleoside triphosphate precursors. One is called ATP, GTP, then UTP, and CTP. You require ATP, GTP, UTP, as well as the CTP. So these are the 4 activated precursors. So what are the 3 components? One is called as a template. Second one is the precursors: ATP, GTP, UTP, as well as the CTP. Then you require an RNA polymerase enzyme. You require an RNA polymerase. So these are actually the 3 components that are being required for the transcription process. Now, the process involves again different phases. So to start, what is transcription? What do you mean by transcription? Transcription is nothing but you can say the conversion of the DNA into the messenger RNA molecule, or DNA forms a messenger RNA molecule with the help of the process called as nothing but the transcription. And the transcription involves 3 components. What are those? One, we call it as the template. Second one are the precursors, and the third one RNA polymerase. And further, this process involves 3 steps. What are those? One is called initiation, second one is called elongation, and the third one you call it as termination. So it involves 3 phases or the 3 stages. One is called initiation, elongation, as well as termination. Now, what is initiation? Initiation actually is the first signal. So the start signals on the DNA, they are called as the promoters. All the promoters are AT-rich regions. They are situated between 10 bases within the initiation. This region has a common sequence of TAATG, and they are commonly known as Tata box. The sigma factor recognizes the start signal on the DNA, and it directs the binding of the enzyme to the promoter region of the DNA. So this is how the initiation will start. So this is okay. The next you have the initiation. So here the start of the transcription will take place at the initiation site, which consists of 1 to 2 bases. The binding of RNA polymerase to DNA will result in local unwinding of the DNA double helix. Then a short segment of DNA will now open up. Synthesis of messenger RNA will begin on one of the 2 strands of the DNA. Then the first base is RNA. In RNA is always a purine, either G or A. This is how the initiation will start. The next, elongation. Now here what happens is when once the initiation starts, the next step actually is being followed by elongation. Now here the chain will elongate, and this elongation will take place by the addition of various ribonucleotide triphosphates, as I have already told you. That is in the form of ATP, UTP, GTP, as well as the CTP. And for each nucleotide added to the RNA chain, a pyrophosphate molecule is also given off, and this is hydrolyzed to inorganic phosphate. The elongation always takes place in 5' to 3' direction. So what are the 3 phases that occur in the protein synthesis? One is called as initiation, second one is elongation, and the third one is the termination. So the elongation process actually takes place by the addition of this ribonucleotide triphosphate. This is how the elongation will take place. The next, this is elongation. So elongation of RNA chains, we can say, takes place by means of core enzyme, which moves along the DNA template. After the commencement of the chain elongation, the sigma factor will dissociate from the core enzyme and it can be released. The last is your termination. RNA synthesis can be terminated by 2 types of mechanisms. The first type, RNA polymerase, reads an extended poly A sequence on the DNA, resulting in an RNA transcript with a terminal poly U sequence. The second type, termination factor or the Rho factor, will cause the release of the transcribed RNA molecule. So this is Rho independent transcription, termination. This is the termination process. Okay. Now the overall basic what is the process of the protein synthesis in the bacteria we'll see now. So we all know actually what is protein synthesis. Protein synthesis is nothing but the synthesis of proteins or the formation of the protein. For this you require the DNA, RNA. So protein actually is not formed directly. But the protein, first the DNA will come into picture. DNA actually will get transcribed into messenger RNA molecule, and this messenger RNA molecule will get translated into the protein molecule. And it involves two phases. One is called as transcription, and the other one we call it as translation. Transcription involves actually the three components. One is called as a template, then you have got the precursors, and then the last one is the RNA polymerase enzyme. And it basically it requires three phases. One is called as the initiation, and then you have elongation, and then you have got the termination. Now we'll see what are the actually how actually it takes place in case of with reference to bacteria. Now, first one is transcription. So transcription actually is a selective process. Only certain parts of the DNA actually are being transcribed. Second, RNA will be transcribed from single stranded DNA normally. So only one of the two DNA strands, that is a template strand, actually is being copied into the RNA, but not both. Only one will be copied. That means it forms a study. Then ribonucleoside triphosphates, they are used as the substrates in this RNA synthesis. So two phosphates are cleaved from a ribonucleotide triphosphate, and the resulting nucleotide is joined to the 3' OH group of the growing RNA strand. Then next, what happens? RNA molecules, they are antiparallel and complementary to the DNA template strand. So transcription always occurs in a 5' to 3' direction, meaning that means RNA molecule grows at always a 3' end. The next, the transcription will depend on RNA polymerase, a complex multimeric enzyme, and it consists of a core enzyme you all know, which is capable of synthesizing RNA and other subunits that may join transiently to perform the additional functions. The next, the core enzyme of RNA polymerases, what it will do, it requires a sigma factor in order to bind to a promoter and then initiate the transcription. The next, the promoters contain short sequences which help in the binding of RNA polymerases to DNA. So these sequences are interspersed, we can say, with the nucleotide that play no role in the transcription. And RNA polymerase again will bind to the DNA at a promoter. It will begin transcribing at the start site of the gene, and then it will end the transcription after the terminator has been transcribed. So this is how actually the the protein synthesis will take place. So we all know actually the central dogma of protein synthesis, or the gist of protein synthesis, is nothing but protein synthesis is what? Protein synthesis is nothing but it is formation of a protein. Why the proteins have to be synthesized? Because proteins are very, very essential. They form the structural component of each and every cell and each and every organism. They play a very, very important role, so that need to be synthesized. They are very, very important to each and every living organism. So how these proteins are synthesized? For this, you require the DNA as well as the RNA. So the proteins are being synthesized from the DNA molecule. First, the DNA actually will get transcribed, and this transcribed DNA will later get translated into the messenger RNA, and this messenger RNA will later get translated into the protein molecule. So that is how actually the protein synthesis will take place. For this, we have seen what are the different phases and what are the different, the components that are actually required for the protein synthesis. Okay, so with this today, now we will wind up the class. Tomorrow again, we will start with the fresh chapter. So today we have discussed about the DNA molecule as well as the protein synthesis.